Hi everyone. Before this lecture, I would like to show the class results from exam 1. The class average is 85, which has been the highest average in all classes I have been teaching so far. We have 4 students who got perfect score. We also have several students who missed only one question. Very impressive. I hope everyone can keep the good work in future exams. Starting from today, we will learn genetics that is no longer covered in general genetics class you learned before. It could be more difficult for some students, so make sure you follow my lectures really well. Also, for students who did not do so well this time, you still have a lot of opportunities to catch up, so don't give up. Today we will discuss cytogenetics, which is the study of inheritance in relation to the structure and the function of chromosomes. You may notice that I have changed the color of word chromosomes into red because this study has to be done at the chromosomal level. Remember, at which stage can we observe chromosomal structure? Yes, only at mitosis. Different organisms have different numbers of chromosomes. This table lists the chromosome numbers in selected organisms. Animal chromosome numbers range from 254 in this is called a hermit crabs to 2 in a species of roundworm. This is a kind of fur called this is a very complicated name, but it has more than 1,000 chromosomes. Humans have 46, and the chimpanzees have 48. And yes, potatoes also have 48 chromosomes. We have learned that chromosomes condense and replicate during cell division. Due to the method scientists use to prepare the chromosomes, most of the chromosomes we saw are this letter X shape. Each X-shaped chromosome has 2 sister chromatids. We learned that and we tested that. These sister chromatids have identical DNA sequences. This is a consequence of what? Yes, DNA replication. We also learned telomeres and centromeres, right? They both have a super condensed and part of the heterochromatin observed in nucleus. Okay, so telomeres can prevent chromosomes from sticking together, and the shortened telomere is a characteristic for cell aging. For example, if we compare 2 cell populations, one from a 70-year-old man and the other from 20-year-old man, you probably can tell who they belong to just by measuring the length of their telomeres. Who will have the longer telomeres? Yes, the young man. After each cell division, the telomeres will be shortened, okay? This is one major reason why when we isolate primary cells from human, the cells can only be maintained in culture for a certain time before they stop growing and gradually die. We call this as a cell senescence, okay? Then you may ask, how about cancer cells? Many cancer cell lines can be cultured in vitro for almost an unlimited time. Interestingly, in cancer cells, the length of telomere is not shortening. This is because in many cancer cells, there's an enzyme called telomerase. It has a high expression level. Telomerase is responsible for maintaining and protecting the ends of the chromosome from degradation. So it maintains the same length of telomeres in cancer cells. Cytogenetics studies chromosomes. So based on their sizes and shapes, scientists characterized them into 3 types. Showing here are these 3 representative forms of chromosomes. On the left, this is the human chromosome 3. this is a large chromosome, and because the centromere is centrally located, it is categorized as called a metacentric chromosome. In the middle here, this chromosome 17 is a sub-metacentric chromosome because the centromere divides the chromosome into two arms of unequal size. On the right, this is a chromosome 21, has a centromere placed very close to the one end, and this is called a acrocentric chromosome. Okay, in humans, the short arm, like here, shorter arm of each chromosome is called the P arm, and the long arm is called the Q arm. The symbol P was chosen from for the short arm because it stands for the word petite, which in French means small. The symbol Q was chosen from the long arm because it is next letter in the alphabet. So this is easier for you to remember. Okay, this slide is a quick reminder that chromosomes can also be grouped into autosomes and sex chromosomes. We have 44 autosomes and 2 sex chromosomes, right? So we learned that. In order to study the chromosomes, scientists would need to know which chromosome is which. The way to differentiate each chromosome is through identifying their unique bending pattern in each chromosome. G banding, or this GIMSA bending, is a technique used in cytogenetics to produce a visible karyotype by staining condensed chromosomes. Heterochromatic regions, which tend to be rich with adenine and thymine, also we call the AT-rich, those kind of DNA, and they are relatively gene poor, that means do not have many genes, stain more darkly in G banding, okay? This picture on the right shows a karyogram of a human chromosome set, showing the distinctive bending pattern of each chromosome. So you will be able to tell who is who. The location of a gene or chromosomal region can be identified using a descriptive address related to bending. Okay, so this is similar to the address we use every day. It's very like our school is in the country of United States, state of Texas, city of Houston. A gene locus contains the number of chromosomes and arms and region and band, okay? So showing over here. So for instance here, we know that in each autosome is numbered from 1 to 22, right? The sex chromosomes are X and Y. Within a chromosome, the short arm is the P arm and the long arm is the Q arm. Each arm is divided into numbered regions. Within each region, the bands are designated by numbers. The area marked with an arrow, for instance, here is a 1Q2.4. So that indicates it's in chromosome 1, it's in the long arm, and it's in the region number 2 and the band number 4, okay? The bands are counted from the centromere and then out toward the telomeres. But in reality, do you think the chromosomes will look like what were shown in previous slides? Not really. A nicely spread chromosomes from a metaphase cell will look like the picture shown here, okay? So based on the bending patterns identified from each chromosome, scientists have developed software to strengthen and arrange the chromosome to what shown here in the right side. So this is called a karyotype, meaning an image of a complete set of chromosomes photographed from a metaphase cell. This figure shows how a karyotype is made in the clinical lab. So usually 10 to 20 ml of blood is drawn from an individual, And the white blood cells, okay, can be cultured in vitro in labs. Remember that red blood cells do not have a nuclei, therefore they cannot be used for karyotype. Okay, so the red color shown in this culture facts is a little bit misleading here. But anyway, so a mitogen here called this fetal hemagglutinin, also known as a PHA, is used to treat the white blood cells to promote the cell proliferation. Okay, we learned in the cell cycle, right? Once the cells start to proliferate, they will enter cell cycle and ultimately enter mitosis. Okay, at this time, the scientists using the chemical named calcimide. They are using calcimide to treat the cells. So calcimide is a chemotherapy compound. It inhibits microtubule formation, therefore disrupts the mitotic spindle formation. So as a consequence, the treated cells were all arrested in the metaphase, all right? So we learned that in metaphase, our chromosomes are all replicated and look like an X shape, right? So nuclear membrane is disappeared in metaphase. The cells are easily broken, and the chromosomes can be stained. After the staining, the chromosome can be examined under the microscope and analyzed. Okay. Karyotyping could tell us a lot of information. It tells us the total number of chromosomes. It tells us the gender of the patient. It tells us if there is absence or additional individual chromosomes. And it also tells us if there's any structural abnormalities of any chromosomes, such as like translocations, duplications, and deletions. We'll discuss these abnormalities one by one a bit later. Although the G banding is a very useful tool to identify individual chromosomes, with the advancement of labeling technology, scientists developed more sensitive ways to stain the chromosomes for karyotyping. We call this as a chromosome painting. Okay, so shown in the figure A here, is a chromosomal painting with 5 different colored markers in a normal cell. Based on various colors of the individual chromosomes, chromosome painting produces a pattern that highlights the translocations, duplications, deletions, and the aneuploidies found in cancer cells, as shown in this figure B. In this picture, you can easily tell that the genome in this cancer cell has been terribly altered, showing all these abnormalities. This table lists the several genetic disorders with chromosomal aberrations. Some of them will be discussed in our group project. Okay, karyotyping can be done in adults using blood samples. We just learned that. But can it be done in fetus? The answer is a yes. The only difference is the different source of cells collected for karyotyping. This picture shows one method of a prenatal chromosomal analysis by karyotyping. This is called amniocentesis. This is a procedure in which the amniotic fluid is removed from the uterus for testing. In amniocentesis, a needle is inserted through the walls of abdomen and the uterus to collect a sample of amniotic fluid. The fluid contains fetal cells that can be used for prenatal chromosomal analysis, similar to what we have shown with white blood cells. This procedure can be performed at about the 16 weeks of pregnancy. At this stage, there is enough fetal cells can be connected, and the risk of miscarriage is relatively lower. Prenatal karyotyping can be done even earlier. There's a technique called cryonic villus sampling technique. They can be performed at 8 to 9 weeks of pregnancy. In this procedure, a catheter is inserted into the uterus through the vagina to remove a sample of fetal tissue from the chorion. The function of the chorion is to contribute to development of the placenta. Cells in the tissue can be used for chromosomal analysis. The previous two methods are invasive methods and may have risk to induce miscarriage in the pregnant women. In 1997, the scientist named Dennis Lowe discovered the fragments of fetal DNA in the mother's circulations, called the free fetal DNA, also as FFDNA. Fetal DNA can cross the placenta and enter the mother's bloodstream, okay? And then isolation of the analysis of fetal DNA in the maternal blood is a non-invasive alternative to amniocentesis and chorionic villus biopsy. Therefore, it has the lowest risk on causing miscarriage. Currently, the fetus has to be at least 10 weeks to perform, and the method has been effective to screen genetic disorders such as Down syndrome, trisomy 13, and trisomy 18. Because of Dennis Lowe's important contribution, a few days ago, he was awarded with 2021 Breakthrough Award with $3 million prize, which is the highest scientific prize in the world. From this example, you can tell that generating good ideas is really important for scientists. Many times, a big advancement of science or technology is simply originated from a good idea. We'll see more examples in our future lectures. Now let's discuss what kind of abnormalities can chromosomes have. First, the changes in chromosome number can cause genetic disorders. Organisms with multiples of the monopoid number of chromosomes are called euploidy. We learned in earlier chapters that eukaryotes normally carry either one chromosome set, we call the haploids, or two sets, we call the diploids. Haploids and diploids then are both cases of normal euploidy, okay? Euploid types that have more than two sets of chromosomes are called polyploidy. For example, triploid, that means 3N, and a tetraploid cell, that means 4N cells, are polyploid. Okay, polyploidy is abnormal and may occur due to abnormal cell division. So aneuploidy is the absence of abnormal numbers of chromosomes in a cell. For example, a human cell having 45 or 47 chromosomes instead of the usual 46, all right? So it does not include a difference of one or more complete sets of chromosomes. So you have to make this difference, okay? A loss of a single chromosome is called a monosomy. In human, monosomy has 45 chromosomes, and a gain of a single chromosome is called a trisomy. A human example, like human trisomy has 47 chromosomes. I'll give you an example of a polyploidy, okay? So this slide shows a triploidy. The karyotype of a triploid individual contains 3 copies of each chromosome. It is a rare chromosomal abnormality in which fetuses are born with extra set of chromosomes in their cells. It happens in 1% of all conceptions, but 99% of the cases result in miscarriage. So only very few live births, and the affected babies usually die within a month after birth. A lot of times, triploidy result from dispermy, meaning one egg is fertilized by 2 sperm cells. In contrast, aneuploidy is most often due to non-disjunction during meiosis. Non-disjunction is defined by the failure of homologous chromosomes or sister chromatids to separate properly during cell division. I'll show you how this happens. But first, let's discuss the aneuploidy that happens on autosomes. Okay, one scenario is autosomal monosomy. Autosomal monosomy are rarely observed in miscarriage or live births, suggesting that this abnormality leads to embryonic lethal at a very early pregnancy stage. On the other hand, autosomal trisomy accounts for half of the chromosomal abnormalities observed in miscarriages, but a few autosomal trisomies can survive and give live birth. Several autosomal trisomies often seen trisomy 13, trisomy 18, and trisomy 21. Trisomy 21 is also known as Down syndrome. It is the only trisomy that allows the survival to adulthood. Let's take a look at what is non-disjunction and how it leads to an aneuploidy. As I mentioned, that non-disjunction is defined by the failure of homologous chromosomes or sister chromatids to separate properly during cell division. Because separation of homologous chromosomes occurs in meiosis 1, non-disjunction in meiosis 1 can lead to aneuploidy. Showing here in meiosis 1, this one pair of chromosomes fail to separate properly at anaphase 1 of meiosis. So leading to these two gametes carrying both carry both members of chromosome pairs. This is called N+1, and two are missing one chromosome, and then show as N-1. Non-disjunction can also occur in meiosis 2, when separation of sister chromatids becomes abnormal. Okay, shown in this slide, the meiosis 1 is normal. Okay, but in meiosis 2, non-disjunction occurs in one cell, this one, producing two cells with abnormal haploid number, one cell with an extra chromosome, and one cell with a missing chromosome. Now let's see several disorders of autosomal aneuploidy. Okay, trisomy 13 is also called Pattern syndrome, which contains an extra chromosome 13. So the total chromosome numbers are 47. In genetics, we use 47 plus 13 to show its cytogenesis. The extra genetic material disrupts normal development, causing multiple and complex organ defects. Trisomy 13 affects 1 in 10,000 births, and the parental age is the only known risk factor. The risk of this syndrome in offspring increases with maternal age at pregnancy, with about 31 years being the average. Trisomy is lethal. Approximately 90% of infants die within the first year of life, and many of them die within the first months of age. These children who do survive past 1 year of life are typically severe disabled with intellectual disability, seizures, and other issues. Another autosomal aneuploidy disorder is the trisomy 18. This is also called the Edwards syndrome, which is caused by the presence of a third copy of or part of the chromosome 18. The disorder affects 1 in 11,000 births, and 80% of cases are females. Okay, babies are often born small and have heart defects. Many of those affected die before birth or within 2 to 4 months of age, and the survival beyond a year of life is around 5 to 10%. Similar to trisomy 13, parental age is a common risk factor for this disease. The infant shown in this picture have this trisomy 18, and here is the karyotype, and it shows 3 chromosomes 18s. Now let's see the most common disorder of autosomal aneuploidy, that is trisomy 21, or Down syndrome as we usually call. Down syndrome is caused by the presence of all or part of the third copy of chromosome 21. It is usually associated with physical growth delays, mild to moderate intellectual disability, and heart defects. The average IQ of a young adult with Down syndrome is 50, but it varies case by case. In the United States, prenatal screening is routinely done in pregnant women because the incidence is rather high for Down syndrome, which is 1 in 800 live births. Individuals with Down syndrome have a higher risk of early deaths than the general population. This is most often from heart problems or infections. Currently, between 4 to 12% die in the first year of life. The probability of long-term survival is partly determined by the presence of heart problems. In those without heart problems, 80% survive to 30 years of age, and about 10% live to 70 years of age. Pretty good. So the picture here is the karyotype of Down syndrome. It has 3 copies of chromosome 21. It is likely that chromosome 21 is smaller than chromosome 13 and chromosome 18. So Down syndrome is not as severe. Interesting, the risk of Down syndrome is also related to the maternal age. Although chromosomal abnormality can be associated with many factors, including like genetic predisposition or exposed to radiation or viral infection, or even abnormal hormone levels, the major and most important risk is maternal age, okay? By studying Down syndrome, researchers have found that 94% of non-disjunctions occur in mother, and most in males is 1, okay? Let's see the correlation next. So this figure shows the relationship between maternal age and the frequency of trisomy, that means Down syndrome, all right? You can see that the risk increased rapidly after 35 years of age. And age of 44 here, about 2% of the babies will be born with Down syndrome. So this is the same with other trisomy disordered. So showing here, this is the frequency of trisomic conception in different ages of mother. By age 42, showing here, about 1 in 3 identify the pregnancy is trisomic. Okay, very, very high. Then you may ask, why is maternity age such a strong risk factor? Actually, you should be able to answer this question based on the knowledge we have learned so far. The major reason is that all the primary oocytes are already formed and resting in meiosis 1 when the woman is born. The longer the oocyte stays, the more chance they will be damaged by intracellular or environmental factors. When the woman gets older, the oocytes may have more damages. That's the main reason. And the second reason is maternal selection. Okay, chromosomal abnormal embryos are typically preventing from implant into the uterus. However, this maternal selection may become less effective as women age, leading to more aneuploid births in older women. Okay, by learning this course, I hope I can convince you that not to have babies too late. Okay, in my personal opinion, before 30 probably is the best. We finished discussing autosomal aneuploidy. Now let's discuss sex chromosome aneuploidy. Interestingly, sex chromosome aneuploidy is more common than autosomal aneuploidy. Both X and Y chromosomes can be involved. But as you may recall, X chromosome is much bigger than Y chromosome and has many more genes. So at least one copy of X chromosome is needed for human survival. As a matter of fact, embryos missing X chromosomes such as 44 minus XX and 45Y are not even observed in miscarriage, meaning any of these defective embryos have been eliminated very early in pregnancy, right? So 44 minus XX means total 44 chromosomes and no any sex chromosomes. And 45Y means total 45 Y chromosomes with Y as the single sex chromosome. All right. So also additional copies of the X or Y chromosome interfere with normal development. This is not the more the better. Okay. So we'll see some examples. Okay. This is Turner syndrome. Okay, Turner syndrome is a monosomy of the X chromosome, meaning that the affected individual misses one X chromosome. Turner syndrome is the only viable monosomy in humans and affects 1 in 5,000 female births. The picture on the left shows the karyotype of Turner syndrome. On the right shows a girl with this Turner syndrome. Turner syndrome can result from meiotic or mitotic non-disjunction. However, it does not seem to associate with maternal age. The affected individuals often have a short neck, low set ears, short stature, and may develop vision and hearing problems. They cannot have babies, but most people with Turner syndrome have normal intelligence. Another sex chromosome related aneuploidy is the Klinefelter syndrome, which is resulted from 2 chromosomes in males. Okay, therefore the affected individuals have 3 sex chromosomes instead of 2 normally. This happens 1 in 1,000 male births, and both maternal and paternal age are risk factors. The primary feature are infertility and small polyfunctional testicles. Intelligence is usually normal. However, reading difficulties and problems with speech are more common. Okay, in this picture shows the characteristic of the karyotype of Klinefelter syndrome. You can see that here has 2 X chromosomes and Y chromosomes. And on the right, this is the baby with this syndrome. And also, this is the picture of when he is getting older. Okay, so actually, this is a young man who has the Klinefelter syndrome. So, male birth. Then would it be possible to have 2 Y chromosomes? The answer is yes. This is named as XYY syndrome. Similar to Klinefelter syndrome, it also occurs in 1 in 1,000 male births. So in the general population, the incidence is approximately 0.1%. However, this rate is much higher in psychiatric and panel institutions. The picture shows the karyotype of XYY male, so they have 2 Y chromosomes here. Here is another slide of XYY syndrome. Affected individuals are usually taller than normal. Many experience severe acne when they are teens. Additional symptoms may include learning disabilities and behavior problems. Intelligence is usually in the normal range, although the IQ on average is 10 to 15 points lower than their siblings. Aneuploidy are major cause of miscarriages and birth defects. For autosomal aneuploidy, only trisomy 13, 18, and 21 occur in live births. The rest are eliminated by miscarriage as pregnancy progresses. The table here shows the chromosomal abnormalities identified in miscarriage. And if you add up these numbers together, then 66% of miscarriages are caused by chromosomal abnormalities. Aneuploidy are also associated with many cancers, especially pediatric leukemias. We'll talk more about those in the future. The frequency of aneuploidy changes dramatically over developmental time. Between 6 to 8 weeks and 20 weeks, you can see here about 35% of spontaneous abortions are aneuploidy. At around 20 weeks, the frequency falls by an order of magnitude to about 4% stillbirths. The frequency decreased again by an order of magnitude, that about 0.3% of newborns being aneuploidy. Therefore, you can tell that miscarriages are not always bad. It is a natural selection to produce healthy babies. And every one of us is a winner as we went through all the selection process and came to this world eventually. Now we finish the introduction of abnormalities in the number of chromosomes. I would like to spend some time introducing structural changes within and between chromosomes. These include chromosomal deletion, duplication, translocation, and inversion. All right. So this is a diagram of what chromosomal deletion and duplication really mean. Pretty easy to understand, right? In a deletion, part of the chromosome is lost, showing here the segment F is lost, right? In a duplication, chromosome segment repeated. In here, the segment DE, they are duplicated. This table lists some variable conditions caused by large-scale divisions. For example, we have learned that this RB gene deletion will result in retinoblastoma. In an inversion, the order of chromosome segments is reversed. Here showing the segment EFG is inverted. In a translocation, parts are exchanged between chromosomes. There are two major types of translocations. One is reciprocal translocation, and another one is Robertsonian translocation. Showing here is an example of reciprocal translocation, where segments of a non-homologous chromosomes are exchanged. Okay, non-genetic, no genetic information is gained or lost from the cells in the exchange, but genes are moved to a new chromosomal location. Another major type of chromosomal translocation is the Robertsonian translocation. It is the most common form of chromosomal translocation in humans, affecting approximately 1 out of every 1,000 babies born. In humans, the Robertsonian translocations occur in the 5 acrocentric chromosome pairs. We just learned that, right? So they, those are the chromosome pairs where the short arms or P arm are very, very short, okay? So these chromosomes are 13, 14, 15, and 21 and 22. Here we use the chromosomes of 14 and 21 as an example, okay? So the two chromosomes break at their centromeres, and their long arm fuse to form a single large chromosome with a single centromere, okay? And the tiny P arm over shown here are usually lost within a few divisions. As the picture shows, the Robertsonian translocation results in 6 types of gametes. When these gametes are fused with those of a normal individual, 6 types of zygotes are produced. Of these, the two of them, one is translocational carrier, and then the other one is normal. They have a normal phenotype. But then another one is Down syndrome shown here, and these three are lethal combinations. Lastly, I would like to introduce two other forms of chromosome changes. One form is called a copy number variation, referring to changes in the numbers of copies of chromosomal DNA segments and the genes that they contain. These changes are not as big as the chromosomal deletion or duplications that can be identified under the microscope. Instead, most of copy number variations were identified through genomic approaches. Copy number variations are defined as deletion or duplication of DNA segments from about 1 kilo base pair up to about 5 million base pairs. Here, the picture shows the relationship between copy number variations, or CNV, in these PMP22 genes and the symptoms of this disease called Charcot-Marie-Tooth syndrome, a progressive nerve disease. Normally, in the normal situation, the PMP22 gene has two alleles in our body. But if one allele has two copies of this gene and makes the total copy number to 3, the CMT can develop. As the copy number increases, the symptoms become more severe. Interestingly, if only one allele has these PMP22 genes and the other allele is deleted, the individual will develop HMPP. It's also called hereditary neuropathy with liability to pressure palsy, which is also a disorder of the nerves. This table lists the human diseases associated with copy number variations. These are also good topics for your group project. The other form of chromosomal changes I want to mention is the fragile sites. Fragile sites are chromosomal regions that are susceptible to gaps or breaks. To date, more than 120 fragile sites have been identified in the human genome. Based on their frequency, fragile sites are classified as common or rare. Okay, common fragile sites are considered part of the normal chromosome structure and are present in all or nearly all individuals in the population. But rare fragile sites are found in less than 5% of the population and are often composed of 2 or 3 nucleotide repeats. We're getting to the molecular details in the future. Fragile sites are often susceptible to spontaneous breakage during replication, frequently affecting neighboring genes. This figure A showing here the fragile sites on the human X chromosome. Okay, sites B and C and D. These are common sites and are found in almost all copies of the X chromosome. However, the A, E, and F are rare sites. Clinically, the expression of A is associated with Fragile X syndrome, the most common cause of hereditary intellectual disability. Okay, so this figure is a photograph of an X chromosome showing a fragile site in this position. All right, so that's all for today. See you next time.